We will be presenting a new method of investigating GPCR signaling by employing recombinant receptors activated by inert synthetic ligands. G-protein coupled receptor, or GPCR, is one of the most important classes of signaling receptors and a subject of constant research. Unfortunately, it is difficult to study the physiological effect of GPCR signaling in vivo due to the presence of varying levels of endogenous ligands. Furthermore, traditional experimental techniques tend to cause systemic changes and hence are inadequate in assessing tissue or organ-specific signaling pathways. To combat these issues, DREADs, or designer receptors exclusively activated by designer drugs, are invented in 2007 by Armbruster et al. It is a class of genetically engineered recombinant GPCRs that are designed to be specifically activated by inert drug-like compounds. DREADs are not affected by endogenous ligands, thereby allowing investigators to control its spatial activation when they are transfected into specific tissues using viral vectors. Two components are necessary for this system to work: an inert ligand and a modified GPCR. The most commonly used ligand is clozapine N oxide, or CNO, since it is inert, highly potent, and has high bioavailability in host. Muscarinic receptors are typically chosen as the basal GPCR template. Site-directed mutagenesis is performed in order to abolish their activity for acetylcholine, the native ligand, while maintaining high affinity for CNO. This is accomplished by converting residue 33 on the third transmembrane domain, a tyrosine, to a cysteine, and residue 46 on the fifth transmembrane domain, an alanine, to a glycine, for all muscarinic receptor classes M1 through M5. With such modifications, CNO is demonstrated to have similar potency to M3 DREADs as acetylcholine to native M3 receptors, allowing DREADs to replicate the effect of native receptors when stimulated. If acetylcholine is added to M3 DREADs and CNO is added to native M3 receptors, severe potency reduction of greater than 40,000-fold can be seen in both cases. One common use of DREADs is to assess the effect of neuron silencing in the brain. M4 GPCRs, which are coupled to GI, are known to induce the opening of potassium channels when activated by acetylcholine, resulting in neuron hyperpolarization and inhibition of neuron firing. Recombinant M4 DREADs, or HM4D, are shown to be just as effective in inducing the silencing of hippocampal neurons when activated by CNO, which is evidenced by the voltage traces of cultured hippocampal neurons. With the addition of carbacyl, an acetylcholine analog, hippocampal neuron control group experiences hyperpolarization, represented by the dip in the voltage trace, whereas CNO did not have any effect. Similarly, in neurons expressing HM4D receptors, CNO addition resulted in hyperpolarization, replicating carbacyl's effect on native M4. Furthermore, the expression of HM4D does not affect native M4 function or baseline neuron activity, since carbacyl can still induce hyperpolarization in HM4D expressing neuron population by targeting native M4. DREADs have now been used extensively to elucidate neuronal pathways in specific areas of the brain thanks to their ability to selectively modulate neuron activity. For example, DREADs have played an important role in Down syndrome research, where selective activation of HM3DQ receptor by CNO in the locus coeruleus of the brain has shown to improve memory. This finding stimulated directed research effort in search of a therapeutic agent that can selectively target locus coeruleus M3. On the other hand, there are also some limitations to utilizing DREDs. Firstly, they're not optimal for experiments that require precise temporal control. CNO acts on the order of minutes, while optical methods of neuronal activation can act on the order of milliseconds. Additionally, it is difficult to control the level and duration of neuronal stimulation produced by DREDs. Hence, next-generation DREDs will focus on resolving these issues by searching for a more optimal inert ligand while improving GPCR design to offer better control. Despite these limitations, the current DRED technology remains a very promising novel method of investigating neurophysiology.
Around our environment and is an important cognitive function with specific neural correlates. Cells that fire in position-correlated fields of activity have recently been discovered in an area of the brain called retrosplenial cortex, or RSC. Paravalbumin, or PV cells, are a type of inhibitory neuron found in RSC and are involved in shaping neural activity. Here we seek to address the following questions. What is the role that PV cells play in RSC during spatial navigation? How does chemogenetic inhibition of PV cells affect neuronal activity in familiar and novel environments? We hypothesize that an activation of inhibitory PV cells in RSC using DREDs will lead to disinhibition, or an increase in neuronal activity following the release of inhibitory mechanisms. We generated a PV PD tomato animal from crossing AI14 and PV Cree mice. Head plate surgery was performed to fix the animal's head during training and imaging, and craniotomy was performed to provide a window into RSC during calcium imaging. The animal then began behavioral training, where they learned to run on a wheel in virtual reality and became familiar with the classroom environment. Once the animal was familiar with classroom, they were injected with AAV synapsin Gcamp6S and AAV2 diode DREDs and began 2-photon imaging. PV activity was then selectively modulated following DRED activation using the agonist J60. Calcium activity was reported each day and analyzed using MATLAB and Python.
Our results indicate that activation of DREDs using J60 has lasting effects at least 1 day post J60 administration. There was a significant increase in the spike rate close to the injection site, supporting our hypothesis that inactivation of PV cells leads to disinhibition of cortical circuits. Intriguingly, we also see that cells become less spatially selective in a familiar environment following inhibition of PV cells, and this trend does not carry over to novel environments. This suggests that PV cells are more so involved in the maintenance rather than the formation of a spatial representation. While pursuing our goal to examine the role of PV cells in RSC during spatial navigation, we were able to successfully record from the same set of RSC neurons across multiple imaging sessions as the animal engaged in navigational tasks. Our results suggest that PV inactivation via DREDs leads to disinhibition demonstrated by an overall increase in neuronal activity at least 1 day post J60, and that this disinhibition led cells in RSC to become less spatially selective in a familiar environment, but not in a novel environment. Finally, we developed viable imaging and training paradigms that can be used to study other inhibitory neuronal populations in future experiments, which can provide greater insight into the complex neuronal interactions that give rise to spatial navigation in the brain.
Neuropsychopharmacology, I'm Cynthia Graber. There's a technology that's proven to be increasingly useful in the past decade. It's called DRED, or Designer Receptors Exclusively Activated by Designer Drugs. The current approach is to create a designer receptor that is unlocked by the drug clozapine N-oxide, or CNO. Researchers have used it to study the function of populations of neurons or of particular pathways in the brains in rodents by using the DRED technique to turn on and off these neurons or pathways. But last year, a paper published in the journal Science called the use of DREDs into question. Some people interpreted the paper as an overall indictment of the DRED technology, though this wasn't the original intent of the authors. And so Gary Aston Jones, director of the Brain Health Institute at Rutgers University, and Stephen Mahler, assistant professor of neurobiology and behavior at the University of California, Irvine, published a paper in Neuropsychopharmacology called CNO Evil: Considerations for the Use of DREDs in Behavioral Neuroscience. Dr. Mahler, first, what did last year's Science paper find?
This new paper was looking at this compound CNO, and this is by a group that had used these DRED receptors previously. they noticed that this compound CNO, it has relatively limited access to the actual brain. And so they did these receptor binding assays in which they labeled CNO with a radio tag, and then they looked at where it ended up in the body and brain. And they found that relatively low amounts of it were actually ending up in the brain, binding to the DREAD receptors as the system is supposed to work. What they did show is that CNO, this is a compound that was actually known for a really long time as a metabolite, a natural metabolite of the psychiatric drug clozapine. And this compound binds to endogenous receptors in the brain at high enough concentrations. And so they found that the clozapine, in fact, was ending up in the brain to a greater extent than CNO was. And their conclusion was that the clozapine actually was the compound that was acting at the DREAD receptor instead of CNO, which is supposed to be an inert drug and have no actions outside of the DREAD receptor. And so this then opens the possibility that this drug is not just acting at the DREAD receptor, but also acting at endogenous receptors in the brain, such as dopamine, serotonin, histamine, et cetera, which then raises concerns of non-selective effects of this manipulation. In other words, we may have sort of like a skeleton key here instead that acts on lots of receptors, doesn't just open up our DREAD receptor lock, but acts at dopamine and other types of receptors that are in the brain that we're not intending to manipulate.
Dr. Aston Jones, what are your thoughts on the finding that CNO is back metabolized into clozapine?
I think an important point is that although clozapine does act at other receptors in the brain in addition to the DREADs, like any drug, it's dose dependent. And so it really depends on the amount of clozapine that gets generated from the CNO as to the degree of problem that back conversion causes. And the DREADs are exquisitely sensitive to clozapine. They have much higher affinity for clozapine than even the CNO. And that was known from the time DREADs were first described by Brian Roth's group. But what's, I think, a little less clear is how much clozapine is available to act at these other receptors. If very little is there, then that's a minimal problem, but if more is there, then that could be a problem.
So what do the results of this paper mean for labs that are using the DREAD technology in their experiments?
The crucial finding of this paper is that CNO has limited access to the brain because its penetration through the blood-brain barrier is relatively weak. And that clozapine that comes from conversion of CNO to clozapine may actually be having effects at the DREAD receptors in a way that was not at least appreciated as much as perhaps it should be. Our point, though, is that although this is the case, that clozapine perhaps acts at the DREAD receptors to some extent in these animal models, the data that come from these models is still interpretable given the proper control conditions, which most of these experiments include. And I think one of our points is to really underline that such control conditions are essential in these experiments in order to interpret the effects of them. But if those control conditions are included, those effects are readily interpretable, and this tool is still a really valuable tool in our toolbox.
It's clear in your paper that you both are absolutely in favor of DREADs and still find the technology incredibly useful. So one suggestion the authors have to deal with the issue they reported is to give clozapine itself in low doses instead of CNO. What are your thoughts?
We've pointed out that there could be some limitations of that. First of all, we would rather just in principle not give a compound that is a known psychiatric drug, even if it's at a dose that is low enough to be ineffective at endogenous receptors. It's theoretically possible for this drug to have some effect, perhaps subtle effect that is difficult to see, but could potentially affect our results and our interpretation of our results. And another possibility is that clozapine itself, it of course is also metabolized like any drug. And so potentially by giving clozapine, the effects of that drug at the DREAD receptor would be shorter acting than giving this compound CNO, which could be construed based on this paper's interpretation as a prodrug for clozapine anyway. So in other words, its actions might last longer because of the fact that it is being metabolized over time and providing steady levels of clozapine over the course of hours.
I would just add, though, that while clozapine could be used as an agonist for the DREAD receptors, There are serious side effects of clozapine that would limit the interest of clinicians in using it. It compromises blood function and causes a lot of cardiovascular problems, or can potentially do that. But there are other agonists that are known now to also have very high affinity for the DREADs, including another neuroleptic called olanzapine, which has many fewer of such side effects. And there are other agonists that have been described as well. So there's a widening array of tools available for manipulating these designer receptors.
And that's the eventual goal, to use these designer receptors and the drugs that can manipulate those receptors in humans.
Because we could express these receptors that themselves have no action. These are new receptors. The cells don't normally have them. And now the cells will start to produce them. We can do this in rodents. We can do this in primates. There's no theoretical reason to think we can't do this in humans. In fact, these same sorts of gene therapy approaches are being used today in humans. So the problem is, what do we do with this new tool, this lock that we insert in these neurons? Can we actually turn the key to correct the aberrant neural activity that underlies these psychiatric symptoms? In order to do that, we need the correct key. And so this is what this controversy is all about. Is our key acting only at our lock, or is it acting at other sorts of locks in the brain?
Just to expand a little bit on the importance of the DREADs and why our paper's main point was to not throw the baby out with the bathwater. The DREADs have enormous clinical potential for several reasons. One is that they're G protein coupled receptors, and many FDA approved drugs that are clinically effective are G protein coupled receptors. So these receptors, these DREAD receptors, are tapping into the same signaling pathways as we already know to be clinically important. Another reason is that these DREAD receptors are human proteins. So the thought is that as far as the outside of the cell is concerned, it would look just like a human muscarinic receptor and would be unlikely to generate an immunogenic response. And there's new receptors and agonists for these receptors being developed all the time. So this whole area, I think, is an extremely exciting new set of tools for not only basic neuroscience, but for clinical therapeutics. And the clinical therapeutic end of this hasn't even begun to be realized yet. People appreciate that it has a lot of potential, but that hasn't even started yet. And I think we're going to see a great deal of advance in clinical neuroscience using these types of tools in the future.
This is the podcast for the journal Neuropsychopharmacology. To read the
I hope to pique your curiosity with a vision to the future of how we might enhance brain function as we know it and possibly change our sense of self. If you imagine the brain as this city, up till now it's been as if we've been looking at the city from space. We really haven't had the tools to do anything beyond seeing what whole city blocks are doing. So you can see what happens if there's a blackout and you lose a whole part of the city. What you actually want to know is what the individual components of the city, what the people are doing, and what's the information that's being transported from one part of the city to the other. Optogenetics is the ability to manipulate individual neuronal circuits, refining our vision, the function of individual circuits and how they relate to different aspects of our behavior and of our personality. I'm going to give you a few examples of what's been done with this technology so far. The basal ganglia is a part of your brain that is damaged in Parkinson's disease. This part of the brain contains many kinds of neurons, and they're all intermingled. If you activate a certain subset of them with optogenetics, you can make a rat freeze up and have difficulty walking. If you activate a different subset, the rats start moving faster. They take light-sensitive proteins from very humble species, such as algae that you find in a pond, and can put them into another animal, such as a rat or a mouse. This gives you the ability to control the activity of your brain cells just by shining light on them. So imagine if we could use optogenetics or similar technology to get the input from an artificial sensor into our brain. In principle, we could not only restore function, we could enhance our current functions. So we could give ourselves ultraviolet light detectors, for example, the way mantis shrimps have. Or we could take an example from fruit flies and give ourselves carbon dioxide detectors, and then we could automatically sense our air quality. When things go wrong with the brain, it's just so devastating. I think one of the hopes for optogenetics is that if it can work in humans, that it might be used as a tool for restoring brain function. There's a lot of ethical issues about enhancing your function and making yourself better. This is what actually the whole discussion after my ideas lab morphed into, which was not about optogenetics or not what we might do, but whether we should do it. So that's a really great point. So would there be a side effect to increasing your capacity by 20%? It's really, really fun to fantasize about the future. You know, in 10 years' time, we might all be enhanced with implants. That's the future, and it's a future that should be very carefully considered. And I think what we have right now, which is just the possibility of learning, is actually exciting enough on its own.
Hello. It is my great pleasure to share my group's latest new technology termed Optogenetics. This is a technology that uses medical ultrasound to produce local, on-demand light emission at any place or any depth in the brain for an optogenetic brain-computer interface. Conventional brain-computer interface relies on invasive electrical brain implants to interrogate and modulate neural activity. Our approach, on the contrary, uses brain-penetrant focused ultrasound to communicate with the brain tissue in a non-invasive manner. Our approach is based on optogenetic modulation of neurons. Optogenetic has its own intrinsic challenges since most opsons are responsive to visible light, which has limited tissue penetration. As a result, an in vivo optogenetic brain interface usually requires invasive means such as the optical fiber implant to deliver light into the brain tissue. In our approach, we have used mechanoluminescent nanoparticles to convert ultrasound, which has deep brain penetration, to light emission, which activates channel rhodopsin for neuromodulation. These mechanoluminescent nanoparticles are oocyte zinc sulfide nanoparticles co-doped with trace amounts of silver and cobalt ions. Zinc sulfide is a direct bandgap semiconductor that absorbs 400 nanometer blue light and excites its electrons in the valence band to the conduction band. Unlike the pristine undoped zinc sulfide that releases the photoexcitation energy immediately afterwards, the excited electron here is trapped by this cobalt dopant until ultrasound kicks in. Besides being a direct bandgap semiconductor, zinc sulfide is also piezoelectric material, which means the mechanical stress which is induced by the ultrasound leads to charge separation and band tilting. Then this tilted band effectively eliminates the trapped state, thus releasing the photoexcitation energy stored in the excited electron. Then through this FRET process, the energy is transferred to the dopant silver to emit blue luminescence at 470 nanometers. This approach would allow ultrasound to control or gate when the blue light should be emitted from zinc sulfide nanoparticles. However, these nanoparticles need to be excited by 400 nanometer light in the first place, as you have seen in the previous slide, meaning once they are in the brain, they can only be triggered by ultrasound once because this 400 nanometer light does not penetrate very deep. To solve this problem, we took advantage of the endogenous circulatory system in the body. Blood vessels are not only present in deep brain for delivering nanoparticles to the ultrasound focus, as a light source, but they are also abundant in superficial tissue where the vessels are shallow enough to recharge the circulating nanoparticles with the excitation light. Therefore, after intravenous injection of these nanoparticles, they can be charged by the 400 nanometer excitation light, which is the purple light here, when they pass through the superficial vessels in the neck and release the energy as localized blue light emission at the focus ultrasound, as the focus of the ultrasound in deep brain. The power density of light emission is about, as you can see from this chart, is above 1.2 milliwatts per millimeter squared, enough to activate opsons for optogenetic neuromodulation. So now, by placing a compact ultrasound transducer on the mouse head, which has no surgical procedure or implant, and focusing the applied ultrasound to the motor cortex, we demonstrated the proof of principle of this ultrasound-mediated non-invasive optogenetic brain interface. So here, the channel rhodopsin expressing mice showed contralateral limb motion, which is in synchrony with the ultrasound pulses only in the presence of the nanoparticles in blood circulation as well as the channel rhodopsin expression, while all other control groups either lacking the nanoparticles or lacking the channel rhodopsin or lacking both did not show any ultrasound-triggered limb motion. Moving forward, we envision the Sound Optogenetics as a non-invasive brain-computer interface that employs brain-penetrant ultrasound alone to read and write neural activity.